What if I told you that we already have technology within our grasp that could cure all diseases? Push the human intellect far beyond the limits of ordinary genius. Bring immortality, or to help populate alien worlds beyond our solar system. I'm not lying, this technology is really here. And if you think it's artificial intelligence, you're mistaken, though not entirely. The ENIAC, the first fully electronic computer. It was constructed in the mid-1940s and was massive, measuring 15 times 9 meters and consuming vast amounts of electricity. Thanks to its complexity, it could perform up to 5,000 operations per second. Remarkable machine for its time, but far from today's standards. The iPhone 13, in contrast, is a million times more powerful machine, capable of billions of operations per second. It achieves this with just a few microchips and a small battery. A stark contrast to the ENIAC's 17,000 vacuum tubes, 70,000 resistors and 1,500 relays. Silicon-based technology is advancing rapidly, and we rightfully take pride in its evolution. Yet, with all our technological advancements, there is a form of technology all around us that predates humanity and surpasses any of our current technological marvels. To grasp its full extent would be similar to encountering an alien civilization capable of advancing humanity forward by thousands, if not millions of years. This remarkable technology is none other than the technology of living systems, biotechnology. In 1953, Watson and Crick shot gamma rays at a crystal of a molecule they believed to be the basic information unit of life. Figure 51, the result of this experiment that helped unraveling the structure of the molecule, DNA, the central element of the incredibly intricate machinery within cells. We often take these processes for granted, but think about it. Right now in your cells, tiny nanobots exist, faithfully replicating your DNA. In almost every cell. They may not look like this, but more like this. Compared to DNA replicas, our iPhones are sticks of cavemen. Creating a similar nanobot with our current technology to replicate DNA is entirely beyond our capabilities. Yet, nature can do that easily. Similar technological marvels with various functions are all around us. In bacteria, viruses, plants, literally everywhere. While we cannot replicate this life technology, we use it for our benefit. Everywhere we look, there are traces of life's technology. PCR tests use nanobots to replicate DNA for disease detection. Antibiotics are created by tiny nanobots creating chemicals to combat bacteria. Genetic engineering uses tiny nanobots that can cut DNA. Gene therapies, modifying white blood cells into cancer-fighting machines. This is biotechnology, a technology with boundless potential. If nature can use nanobots for limb regeneration in some animals, why not in humans? If nature can use nanobots to combat aging, why not in humans? These are the limitless possibilities I refer to. Yet, like with the alien technology, our intelligence remains the limiting factor. We could, in theory, create nanobots doing exactly what we want, but we just don't know how. The nanobots of life are extremely complicated systems beyond the comprehension of human intelligence. But what about using non-human intelligence? Life technology is the technology of 3D space. All molecular nanobots are small proteins that are composed of a sequence of amino acids. It's kind of like Lego blocks that fold into different shapes according to the rules of nature, which directly affect their function. A virus can infect a cell because its surface protein fit precisely into receptors. DNA replicase fits precisely on the DNA and so can grab it and replicate it. Dopamine receptors have a shape that dopamine fits precisely into, and so you can feel pleasure when you eat food. By understanding the 3D structure of the molecules, you are able to control their function and take full control of biological technology. Hi, this is just a reminder that this is a new YouTube channel, so if you like what you're seeing, don't forget to subscribe, comment, share the video, and spread the word. Thank you very much, and let's continue. The problem is how to reliably predict and design the 3D structure. Even a relatively small protein made up of 100 Lego bricks has an incredible number of possibilities for the final shape. Many times there are more possibilities than atoms in the entire observable universe. This task is beyond the capabilities of human intelligence, but not the artificial one. Until recently, the structure of proteins was determined by a difficult and expensive process called crystallography. Determining the 3D structure of a single protein can take many months and cost hundreds of thousands of euros. But in 2018, a huge breakthrough has been made. Since 1993, a prestigious event known as the Critical Assessment for Structure Prediction, CASP, has taken place every 2 years. In this scientific competition, teams of experts were tasked with predicting the three-dimensional structures of proteins based on their amino acid sequences. In 2018, CASP-13 marked a significant milestone with the debut of AlphaFold, a groundbreaking artificial intelligence system developed by DeepMind. AlphaFold caused quite a stir as it outperformed other teams. While the second best team could only correctly identify the structure of 3 out of 43 most challenging proteins, AlphaFold successfully identified 25 of them. In 2020, AlphaFold 2 was opened to the public. Within 18 months, more than 1 million researchers, including virtually all of the world's leading biomed labs, had gained access to it. Previous experiments added the structure of about 190,000 proteins in the European Bioinformatics Institute's database, which is about 0.1% of the known existing proteins. DeepMind uploaded approximately 200 million structures at once, representing nearly all of the known proteins. Of course, AlphaFold is just the beginning of the revolution. Imagine that just like we have ChatGPT today to create text, DALL-E to create images, we could have a generative artificial intelligence for nanobots of life. Generate an amino acid sequence that folds into an enzyme that can find the HIV virus in the DNA and cut it out, effectively curing the patient. Sci-fi? Yes, but maybe not for long. In 2023, researchers at the University of Washington used a deep learning model to create an enzyme that can break down the molecule luciferin and produce light. This model was trained on natural enzymes called luciferases, commonly found in certain kinds of jellyfish or fireflies. But it didn't just copy existing enzymes, but similarly to AIs creating images, the AI used existing data to make 7,500 new enzymes that don't exist in nature. The result was LuxSid1, an enzyme that's brighter and more efficient than any natural one. A huge advancement that wouldn't be possible just a few years ago. The potential for generative AI in biotechnology is limitless. Alzheimer's disease is characterized by aggregation of waste proteins in the brain that are thought to contribute to brain degeneration. Perhaps one day we might be able to generate nanobots that can accurately recognize waste proteins, break them down, and prevent the spread of Alzheimer's disease. Today we can genetically modify patients' white blood cells to target and eliminate cancer cells. Future advancements coupled with AI could lead to even more effective treatments, potentially eradicating cancers entirely. However, these efforts extend beyond medicine. In 2016, scientists discovered Ideonella sechensis, a bacterium equipped with PET nanobot capable of breaking down PET plastics, a novel material for nature. Within just a few decades, evolution has developed a tool to utilize PET plastics as an energy source. The breakdown isn't flawless, as it still produces some harmful chemicals. But if blind nature can find ways to address plastic decomposition, imagine the possibilities with guided design using advanced artificial intelligence. Bacteria with the ability to efficiently capture carbon dioxide from the atmosphere can play a crucial role in addressing the climate crisis. Genetically modified crops have the potential to alleviate famine in impoverished regions of the world. As we advance in scientific knowledge and technology, we will be able to design our own nanobots and eventually entire organisms. We are nearing a point where we can become the architects of biodiversity and masters of life. However, also masters of death. At a pharmaceutical company in North Carolina, a team of scientists is using generative artificial intelligence to accelerate drug development. By inputting extensive drug data and desired properties, the system can suggest more efficient designs for new drugs. But one day they said, what if we flipped the logic and instead of medications, we design poisons? Within hours, artificial intelligence had designed 40,000 chemical weapons, many of which were arguably more toxic than Novichok or VX. The researchers themselves were surprised by how easy it was.
A lot of the things we used are out there for free. You can go and download a toxicity dataset from anywhere. If you have somebody who knows how to code in Python and has some machine learning capabilities, then in probably a good weekend of work, they could build something amazing like this generative model driven by toxic data sets.
The more accessible technology, the greater the risk of misuse. Take the example of the nuclear bomb. Making a nuclear bomb is a difficult process that cannot be replicated in someone's garage. But imagine it would be possible. There would be instructions on the internet on how to make massive centrifuges using a 3D printer to process uranium, which would be commonly available in the supermarket. You follow a simple PDF file step by step, and at the end of the process, you get a dirty nuclear bomb that can blow up half of your city. A picture of a dystopian future, but we are facing something similar on the ground of biotechnology and artificial intelligence. Synthesis of chemical weapons, terrorist attacks by synthesized pathogens, threats are absolutely everywhere. For approximately 20,000 euros, you can purchase a DNA sequence synthesizer, a machine where you can input the sequence and it produces pure DNA according to your specifications. Until recently, it was virtually no problem to download the whole sequence of H5N1 virus, a virus with the potential to kill up to 50% of infected people. Just imagine a fanatical terrorist group had synthesized and modified it. I'm not saying it would be completely trivial, but even 15 years ago, it would have been absolutely impossible. What will it look like 20 years from now? Although progress has been fascinatingly fast so far, we have still managed to effectively contain the risks. We first invented cars and then we introduced seatbelts, discovered dangerous pathogens and later established safety regulations in biology laboratories. Similarly, we initially learned how to code and create malware and subsequently developed anti-malware softwares. With the new technology, this is always like that. We develop it, identify negative effects, and then create effective countermeasures. However, the more powerful technology, the more reckless this kind of approach becomes. On the night of January 24, 1961, a B-52 bomber fell apart during flight over Goldsboro, North Carolina, with 2 nuclear bombs on board. During the crash, both bombs separated from the aircraft and fell to the ground. Although the bombs had several safety mechanisms, on one of them, almost all of them failed, and was only one mechanism away from detonating. We got lucky, as on at least 3 other occasions when similar incidents occurred, and by some stroke of luck, there was no detonation. We have potentially much more powerful technology in our hands today. Are we going to rely on chance and luck again? Or perhaps it is finally time to start taking proactive measures, both in biotechnology and AI. Because if we don't, we may wake up to a morning when seatbelts have no one left to save. I'm Peter, and this is BeWise.